Good evening, everyone. I'd like to welcome you to the National Fragile X Foundation's webinar. Tonight, the topic is Working to Restore Balance to Brain Function in Fragile X Syndrome. It will be presented by Dr. Craig Erickson. I'm Jane Dixon Weber, the Director of Education and Support Services at the Foundation. Before I introduce our speaker tonight, I'd like to first thank Warren Global Conferencing. They've made this webinar possible. A couple housekeeping issues. We will go through all the slides and then take questions. Use the chat box on the right side for your questions, and also use the chat box if you have any problems. Now, I'd like to introduce Dr. Craig Erickson. He's the Director of the Fragile X Research and Treatment Center and the Associate Professor of Child and Adolescent Psychiatry at Cincinnati Children's Hospital Medical Center. He provides clinical care for persons of all ages with Fragile X syndrome. His research focuses on targeted treatment development in Fragile X syndrome, including work to develop biomarkers for use in Fragile X treatment studies. Welcome, Dr. Erickson.
Well, thank you very much. Hopefully my audio is working. If not, I'm sure someone will let me know. But this is really a great opportunity. I don't intend by any means to utilize the entire hour with the slides. We'll have significant time for questions related to this slide set, and I'm happy to answer anything related to clinical trials and new treatment development in Fragile X. I really come to tonight's webinar representing a new group supported by the Foundation, the Clinical Trials Subcommittee of the Fragile X Clinical and Research Consortium, where I serve as the chair of that committee. So really our goal is to bring webinars to all Fragile X stakeholders regarding any new clinical trials, clinical trial ideas, or development activities focused on new treatment development in Fragile X. So we hope that this is really the first of a series of these sort of webinars to provide information. And I also want to make it clear that tonight's content was developed in collaboration with Neuren Pharmaceuticals, who make this compound NNZ-2566, and the Fragile X Clinical and Research Consortium Clinical Trials Committee, which includes broad representation from Fragile X clinics around the country and members of the National Fragile X Foundation. So I essentially am giving this report on behalf of our committee. All the content's been vetted and looked at by all of those members. So with no further ado, I'll get into tonight's topic, which is specifically discussing this compound, NNZ-2566, which is being developed by Neuren Pharmaceuticals for potential use in Fragile X syndrome, and that is currently in a multi-site national clinical trial in Fragile X. So that's our topic tonight. And first, my potential conflicts of interest. And I think my primary conflict of interest is that Cincinnati Children's Hospital is one of many sites for this trial. So essentially, we enroll individuals impacted by Fragile X at Cincinnati. And like any clinical trial site that enrolls individuals, my hospital is paid on a per individual basis when folks are enrolled. So I just want to be clear and transparent about that. We also participate in many other clinical trials that are our own studies and studies from other industry and different partners. So I just want to be clear about any potential conflicts that I may have. So first, I want to just give some general thoughts on the state of novel therapeutics development in Fragile X right now and what our trial committee is doing as an entity. And really our goal is to support new and innovative research focused on treatment outcomes in Fragile X as broadly as possible. So we exist to embrace industry, academia, any others that are interested in developing treatments, not only medications, including potential non-medication interventions in Fragile X syndrome. Our goal is to embrace this work and try to advise and support the development in any way possible, such as having webinars like this that essentially get the word out. We have broad membership across a number of Fragile X clinics, Fragile X clinical and research consortium programs around the country, and a number of members of the Fragile X Foundation are involved. And again, these webinars are just one of many aspects that we're doing to promote research in this area. And this is all reviewed by the entire committee. So where has the field been? And I know that our participants, stakeholders, families on tonight's webinar, many folks know this as well as anyone, in particular if you participated in various trials. You know, there's been significant hope and promise in this field for new targeted treatments being developed in recent years. You know, there have been a number of trial failures recently that we're all aware of. Compounds from Roche, Novartis, Seaside, Therapeutics. And really, in my mind, what we're doing at this point is taking those lessons learned from previous failures and using those lessons to design better future trials, to target new mechanisms of action of drugs, to really move things forward. And I think, you know, it's an understatement to maybe say that some of the results have been less than breakthrough study results. But, you know, there have been some disappointments, and we want to be frank about that and continue to move things forward. And I remain optimistic. Our committee remains optimistic. Those that we partner with at the foundation remain in a similar sense. So, you know, hope still springs eternal. We understand that there's weariness inevitably setting in, given some of these failures. But, you know, what we're going to talk about tonight is a new novel mechanism of action, not related to previous trials. And I think, you know, not only what we're going to discuss tonight, but there are several other compounds in development at various stages that hold a great deal of promise still for significant improvement for individuals impacted by Fragile X syndrome. So again, we're moving beyond the past. We want to learn from the past. We have new novel approaches to outcome measures in the field. We're going to talk a little bit about that tonight related to the NNZ compound from Neuren. We believe, you know, as scientists in the field and researchers and partnering with others, that, you know, broader mechanisms of action may be extremely therapeutic. As we learn more about the Fragile X protein and the broad effect it has on the brain, we may need to take a broader approach than just targeting a single neurotransmitter, which has been done in several previous trials that unfortunately have not had widespread positive results. There are still, though, many questions that we're working to resolve, such as, you know, how long do you need to treat? At what age do you need to treat individuals to show significant impact? We continue to work through that. But I really remain steadfast in my belief in the field that we're still poised for a breakthrough, and we're well ahead most, if not all, other forms of developmental disability new treatment research. So Fragile X is still on the cutting edge. The interest is still there around the world in improving outcomes with new potential pharmacologic and even non-drug treatments. So I think we want to really keep the momentum going, and we want to show the world, that the Fragile X community, that all stakeholders, you know, can really learn from clinical trials, move things forward, can execute trials, and continue to take additional shots on goal to really have a breakthrough. And I think it's with that in mind, you know, that we bring forth information tonight as one such potential option in new development. So a little bit of background on this compound, NNZ-2566. And, you know, drugs in earlier stages of development, they always have these monikers, NNZ-2556 and 66, and then they get renamed. That's just kind of par for the course. So I'll refer to it tonight as NNZ for the sake of simplicity. So we need to understand a little bit about insulin growth factor, because NNZ is essentially an analog or an insulin growth factor-like compound. So this is a growth factor that's released by the liver in response to a growth hormone in humans. It crosses the blood-brain barrier, which means it easily gets to the brain. It's expressed specifically in brain cells, and it helps the central nervous system, or your brain and your spinal cord, grow and develop over time. It promotes survival of brain cells, and synaptic, which are brain connections, to mature as brain cells develop, and plasticity, which is the very important process of laying down new memories and brain adaptation to experience. Meaning, I hear something, I see something, my brain cells lay down new connections, and that's called a plasticity, or a sort of a active response based on stimuli. So that is in part mediated by insulin growth factor and other growth factors that facilitate that. And that's important to really think of in Fragile X because we know that that connectivity is not normalized and is dysregulated in the disorder. In Fragile X, we have some information from animal models that these IGF-1 or insulin growth factor levels are likely decreased. So there's a theoretical basis to want to increase insulin growth factor activity in order to correct potential brain deficits in Fragile X syndrome. So with that said, NNZ specifically is an analog or sort of a synthetic version of something called glypromate, which separates from insulin growth factor in the brain and really is an active part of what insulin growth factor is doing to help the brain develop these connections and grow appropriately. So essentially, we have a synthetic version of this GPE or glypromate, which is an essential part of insulin growth factor. And the NNZ is, as a synthetic analog or a drug that's manufactured, has a longer half-life, meaning it stays around longer, so it has more effect as a drug in that way. It's very stable, and it can be taken orally and still have an active brain effect. So it's able to then potentially substitute for low brain production of insulin growth factor in Fragile X or other disorders in which NNZ is studied. So, we have a synthetic drug developed to sort of replace deficient insulin growth factor 1 activity, potentially in Fragile X syndrome. And then it has the proposed name, getting away from the numbers and the letters, of Trofinetide, which, we have no idea where that name comes from. There are probably focus groups and other things involved. But, we'll call it NNZ at this time and continue to move forward. So what else has this drug been studied in? And I think there's an important potential parallel in a disorder called Rett syndrome, which is another genetic cause of developmental disability. It commonly is also associated with autistic features. And Rett syndrome, in many ways, is a more severe developmental disorder than Fragile X syndrome. It occurs in females. Males who are thought to develop Rett actually tend to not live, essentially to be born, and may well die in utero. Folks with Rett have very severe cognitive and intellectual disabilities. There are no effective treatments. They have a lot of abnormal movements. They tend to lose skills, so they may develop and then lose skills. So it's a very severe disorder. NNZ has been studied in Rett in 56 individuals in a placebo-controlled trial, and in mainly an adult trial with an average age of 25 years. There was a month or 4 weeks worth of treatment at 2 different doses, a 35 or a 70 milligram per kilogram of body weight, twice daily dose. And in the Rett syndrome trial of this compound, both drug doses were very well tolerated without significant safety concerns. And the higher 70 milligram dose exceeded what were called pre-specified criteria for treatment response. Basically, Neuren Pharmaceuticals came up with a list, submitted it in a sealed way to the FDA and others before the end of the trial, and said, across several different scales utilized in Rett syndrome for improvement, this is what we're looking for. And they saw that more so with the drug treatment than placebo in this 56 subject study. And the impact was rated by both caregivers and clinicians in the trial. So, unlike a lot of recent clinical trials in Fragile X syndrome, this is a drug, NNZ, that's actually been studied in another disorder prior to Fragile X syndrome. And a lot of the previous Fragile X work, like Roche and Novartis, there wasn't a sort of track record in developmental disorders prior to work in Fragile X. But that's different here, where we have a track record in another genetic form of developmental disability. So how is this drug relevant to Fragile X? Well, again, it's important to think about the animal model, primarily the mouse model of Fragile X, where it's been noted that the insulin growth factor, or IGF-1, is deficient potentially in Fragile X. So again, the hypothesis or theory of increasing the function has a rationale. And there's been rescues of the Fragile X animal phenotype, both behavior and chemical aspects of brain activity in the Fragile X knockout mouse that are rescued with NNZ treatment. So both positive behavioral changes and correction of some chemical signaling have occurred in Fragile X mouse study, and this is in the published scientific literature. And I'm going to talk a little bit more about the specifics of those results. So first, what does NNZ potentially do in Fragile X in the brain? So here, and hopefully my arrow can be seen, NNZ is known to affect outside of the cell, and where we are here is outside of the brain cell, cytokine levels, which are chemical messengers, that hit a number of different receptors. And essentially, we have 4 receptors here on the slide. So it has broad effect across a number of different receptors controlling activity and signaling that then leads to broad effect inside the cell, where we know in 2 main areas the drug in the Fragile X mouse model normalizes some abnormal activity. And here where I'm circling right now is something called ERK, or extracellular signaling kinase. This is a chemical signaling pathway inside cells, in particular brain cells, that's known to be upregulated in the Fragile X mouse. And this is known to be reduced and normalized with NNZ treatment. So we have a rationale that what's happening on the outside of the cell with NNZ broadly on signaling is having some internal cellular normalization in the Fragile X mouse model. We see the same thing in the mouse model with AKT, which is another cellular signaling pathway that's also normalized. So I think the simple concept here is for glia cells, which support brain cell development, to restore function at these receptors, a number of different receptors, leading to internal brain cell activity that normalizes. And we're going to talk about some of this internal activity that normalizes specifically in the Fragile X knockout mouse. And what happens is by normalizing the chemical activity inside the cell, it leads to appropriate cellular growth and connections and development that we'll show through some of the mouse model data. So now we'll just say a little bit about what are called microglia and astrocytes. These are additional cells in the brain. They have immune function. They protect and support neurons or brain cells. They help manage connections. And they normally rely on and produce insulin growth factor. So there are some hypotheses that these cells' activity may be involved in the dysregulation of insulin growth factor in Fragile X, and this is again being normalized and supported by NNZ activity. So I sort of point this out because it's a novel mechanistic approach to targeted treatment in Fragile X, clearly different than what we've seen so far with selective metabotropic glutamate receptor type 5 and selective GABA-B modulators in the past. So moving on to the extracellular signaling kinase and the AKT. These are the 2 of these sort of inside the brain cell chemical signaling molecules. They're known to be overactive in green in the Fragile X knockout mouse. They're both overactive or dysregulated. And this normalizes, it comes to a normal activity level when you treat the knockout mouse with NNZ. You go here from a high level down to a normal level that's equivalent to mice that don't have the Fragile X knockout. This is the phosphorylated extracellular signaling kinase. The same thing happens with phosphorylated or activated AKT in cells. It's elevated here at baseline, and it normalizes or is reduced with NNZ treatment. So we show that whatever NNZ is doing at a cellular brain level, we can detect inside the cell that chemical signaling pathways are normalizing. And this is important to what we're going to see here in a second, which is that brain cell connectivity is restored in the mouse model with NNZ treatment. So these are some highly enhanced images of individual neurons and what are called sort of dendrites that come off of neurons. And at the top here, we have wild-type or non-Fragile X mice. So what you see is a long neuron or brain cell shaft with a few different connections coming off. In connections, you can kind of think in a simple way is how the brain lays down new memories. So brain cells connect with each other, and then you have this incredible pattern of connectivity. Now, what do we see in the Fragile X mice is the same thing we see in Fragile X human brain samples in folks that have passed away. It's hyperconnectivity, way more connections than you would want, creating static and an inability to lay down the appropriate connection. So this likely may lead to, hypersensory responses and inability to lay down memories and learning in a, quote, otherwise normal way. It's too many connections. So what do we see at the bottom? When you give the Fragile X mouse the NNZ drug, you see a reduction in this hyperconnection, this hyperconnectivity. So in the second from the top, we have hyperconnectivity in the mouse. When that's treated down at the bottom with the NNZ drug, you see a restoration or restoring an appropriate number of connections consistent with the first bar and the third bar that are the mouse that doesn't have the Fragile X knockout. So we restore the appropriate connections and reduce all of these hyperconnections by treatment with NNZ in the mouse. So this is kind of the, meat and potatoes of microscopic brain changes you see with the drug, and it likely relates to those inside the cell signaling changes that I talked about in the prior slide. So basically, brain connections are normalizing with treatment in the Fragile X knockout mouse with NNZ use. Spine numeracy is just a fancy way of saying the number of connections that we saw in the previous picture. Basically, these connections are massively increased. There's more of these spines coming off brain cells. There's an excessive number, and that number goes down and normalizes with NNZ treatment. And that's been published in the medical literature. So we see normalization of all of these branches that are coming off with treatment. There's also some positive behavioral change that's been shown with NNZ in the Fragile X knockout mouse regarding aspects of anxiety, social function, and hyperactivity. Here we see some abnormal social behavior, that can be part of maybe the social anxiety phenotype in Fragile X that's normalized. It goes from an excessive level here in the clear bar with the red outline to a normalized level with NNZ treatment, and by normalized, it kind of comes down to the level of animals that don't have the Fragile X knockout. So what do we see overall in the mouse models? We see increased insulin growth factor levels in the brain, which are low in Fragile X. We see normalization of this inside the cell signaling with the ERK and the AKT, which are known to be abnormal in Fragile X in the mouse. And then we see normalization of this brain cell branching in connectivity with NNZ treatment. So on several different levels, we're seeing NNZ having a positive effect in the mouse model of Fragile X. So now I'm going to talk about the human trial. That's a lot about the background of sort of why NNZ is being studied. So what's going on in humans with Fragile X right now? Well, first of all, clearly this work is sponsored by Neuren Pharmaceuticals. It's manufacturing and developing this compound. They're sponsoring right now a 10-week study. It's a blinded, meaning placebo-controlled study. Physicians, patients, families do not know whether an individual is assigned to the active drug or a non-active placebo solution. It's a liquid medication formulation that's a strawberry-flavored liquid, and you basically either get the active drug in a strawberry-flavored liquid or you get placebo that also ostensibly has a strawberry flavor. Folks are randomized to either 2 doses of the NNZ drug, the 35 milligrams per kilogram of body weight, and a kilogram is about 2.2 pounds, or the 70 milligram per kilogram of body weight twice a day, at least 8 hours apart over the course of the project. It's only involving males right now, ages 14 to 40 years of age, but I have information that likely the trial is going to go down to age 12 in the near future and will additionally enroll 12 and 13-year-olds. But as of this moment, it's age 14 to 40. Individuals have to have some degree of clinical symptoms and be impacted in a way that we would think that clinical symptoms and behavior would change. So with that said, it's possible that some of the least affected individuals with Fragile X syndrome could potentially not be eligible for the study. But in my experience and in talking with my peers, or really I think the vast majority of individuals meet the criteria of clinical symptoms to get in the study. You can't be on more than 3 psychotropic or behavioral medications, which is pretty standard for trials. You have to have stable behavioral therapies for at least 4 weeks. So you can't just be initiating a new non-medication treatment that may confound or make it difficult to interpret potential response. For those that have seizures or epilepsy, they have to be stable, so no active seizures in at least 3 months. And folks with significant medical problems may not be able to participate. And this is true of all trials. If you have major other organ system disease or concerns, it probably makes the most sense to not be in an investigational medicine study. There are a list of excluded medications that are, to a degree subject to change. You can see the list there. It includes a lot of other medicines in targeted treatment studies, some medicines that affect glutamate and GABA activity in the brain. In some situations, these are medicines that individuals could stop or come off of prior to being in the trial if that would be clinically the right thing to do. And we'd always recommend that folks talk to their prescribing provider, physician about that if they're interested in stopping something to enter any trial. So there are 8 visits in the project. Screening includes completing forms completed by the study physician and the caregiver, a physical exam, a heart rhythm strip, blood safety tests. The duration of the visits is variable, from around an hour to several hours, depending on the individual visit. And there's what's called a baseline visit, or sort of the day you first, right prior to taking the study medication, either active drug or placebo. And then you see there are visits every couple of weeks thereafter, including a follow-up well after the treatment period. The study physician and the caregiver are going to be filling out forms about behavior at each visit. There are physical exams frequently and heart rhythm strips, EKGs each time. Safety blood tests and blood tests that potentially look at some of these chemical markers I mentioned that have been studied in the mouse are also studied in the human. We measure cognition and functioning and look at eye tracking, or where individuals with Fragile X may look, whether they look more at eye regions, mouth regions, those sort of things. And then there's also a language task to assess, are we changing communication in any way with treatment. A couple of safety visits on there where there's a home visit option where you don't necessarily have to come into the clinic, so you can keep up every 2-week visits and get a blood test and your vital signs checked, even potentially at home in between. What are potential risks? I like to say, and it's an honest approach, that there's never been a good medicine developed, in particular in neuroscience, that doesn't have potential risks. So this is no different. Chance of headaches, dizziness, potential pain, tiredness, upset stomach, all relatively rare risks. Bruisedness or irritation of the skin during blood draws is a risk. And, there are other potential rare risks that are discussed in detail during informed consent in any trial, and this certainly holds true. The data in Rett syndrome was generally very well tolerated, and, as a study physician involved in this trial, I really take that as a positive when we look at the safety profile. But certainly, you want to be aware of all potential risks if you're considering the project. So what about assessments? You know, there are a number of different assessments, some traditional checklists folks may or may not be familiar with, the aberrant behavior checklist, child adolescent symptom inventory, an anxiety scale. You know, there's an interesting measure for caregivers and really the clinical providers too of, you know, most common concerns. So, you know, as a family with a child or, you know, an adult child with Fragile X, what are your biggest concerns and how do you rate them and do they change with treatment? I think it's really trying to become individual-centered when looking at potential outcomes, and that's included. Neuren is developing kind of a Fragile X specific rating scale where we go through a lot of different domains common to Fragile X, repetitive behavior, social anxiety, repetitive movements, inattention, hyperactivity, you name it, all elements are covered by an assessment and gone over with a clinician. We also look at adaptive behavior, so sort of functionality, how is someone able to take care of themselves, what are they able to do, you know, on a daily basis functionally. And then we measure cognition with a computer-based metric called the KITAP, an expressive language sample. So again, how is someone able to communicate when another individual is prompting and talking to them? And then we do the eye tracking to look at what's gaze like, because we know individuals with Fragile X have some eye gaze abnormalities. They're less likely to look towards eye regions, among other aspects of eye gaze that are abnormal. And then there's a very broad safety approach with physical exams, safety blood tests, EKGs, vital signs, medication diaries. We look at potential adverse effects via a questionnaire. And I think that that is all, you know, done in the sense of safety and very thorough. So what are the main outcomes at this stage of development with NNZ and the current clinical trial that I just mentioned? Really, safety and tolerability is number one at this stage. And then looking at a number of potential behavioral changes, repetitive behavior, speech and language change, anxiety, sensory reactivity, social withdrawal, social cognition, attention, adaptive behavior. At this stage of the game of first study in Fragile X, in humans with Fragile X, I think a lot of the point is really to figure out, you know, if we're seeing a positive signal, exactly where is it? And that's why we have a broad sense of potential outcomes in those participating in this trial. We want to also look at dosing, and that's why there's two different doses to see if, you know, high versus low dose makes a difference with tolerability or with potential clinical effect. So I want to say here near the end a little bit on a subject level assessment, which is kind of a new way of conceptualizing outcome in trials. And it's really trying to look at positive treatment response. It may be different in different individuals. You know, even in a single gene disorder with Fragile X, I think we all know who have family members who are affected or as clinicians working with the population, that there are a lot of individual differences. So I think this methodology really tries to include some specific Fragile X ratings and ratings of issues and most concern to individual families. It shows success as an approach in the Rett syndrome model with NNZ. And it's an alternative to kind of a one-size-fits-all checklist approach that maybe has posed some difficulty in prior trials, like the aberrant behavior checklist or specific other scales. So it's a way to really individualize movement with a trial. So in other words, you know, you could potentially have an effective treatment for Fragile X that may help one patient, one individual with their significant inattention and hyperactivity. It may manifest positively with another individual, more with anxiety. And maybe it even helps a third individual with aspects of self-injury and physical aggression because the manifestations of the disorder may be a little different and unique. So I think this individualized subject-level assessment approach, which is shown to be a technique that's worked in other disorders such as heart failure and other areas, may be a more individualized way to look at outcomes. And I think Neuren as a company is really open to different and creative approaches to looking at outcome measures in Fragile X, and I think this is one way to go about doing it. So what are the sites right now? And there's a number of them. Again, as I said, Cincinnati Children's Hospital is a participant. So obviously we're involved and have that potential source of bias. There are sites across the country. I know several of these sites are newer sites that have been brought on to try to help expand recruitment. I'd also encourage any potential interested stakeholders, family members, anyone, to look up the study, to look at the study website, or look at clinicaltrials.gov, which is really a government-sponsored, objective clearinghouse for clinical trial information. If you go to clinicaltrials.gov and you search either that long NCT number, or if you just put in NNZ and Fragile X in the search box, it'll get you to the trial. It'll have more detailed information about the outcome measures, about some of the specifics, and all the contact information for various sites will also be provided at that clinicaltrials.gov webpage. So I think that's an important objective resource for information. Here's information on some acknowledgments. Neuren Pharmaceuticals obviously has provided a lot of the background information and some of the descriptions in tonight's presentation. They're very open to interface with Fragile X stakeholders at every level to be contacted and discuss aspects of their development program. And then our Clinical Trials Committee, which again has broad representation. We continue to add members from Fragile X clinics around the country and members of the National Fragile X Foundation that are interested in promoting clinical trials and clinical research in Fragile X. So finally, there's my contact information. I'm always open to talk with any families, anybody else. Contact me directly, email our Fragile X program about this or anything else. We really want to be open and help facilitate changing the outcome in Fragile X. And that's a mantra we really feel to the core and want to move forward with as many partners as we can find. Any questions about our committee or interest in interfacing with our committee on any level, you can contact Jane with the foundation. And we're a newer committee in the last 4 to 6 months, and we're ever evolving, and we're open to input, advice, and broad participation. So with that, in our final 20 minutes, I think we're on a good pace that I'll be able to answer a number of questions and continue the dialogue.
Thank you, Dr. Erickson. That was a wonderful explanation of that new drug. I don't know if you can see the questions on the side, but we've got a lot of questions coming in. So would someone who's nonverbal be disqualified from this study?
No, there's no specific disqualification for nonverbal status.
Okay, and will this ever be expanded into girls?
To be honest, I can't answer that question at this time. All I can say is I have not received an indication that the current study in its form has an intention to add females with Fragile X at this time.
Now, are there any preliminary results with regards to, are you seeing any, are the younger ones doing better on this than the older ones, or do we have any preliminary results at this point?
In trials like this that are double-blind in nature, essentially I don't know, the families don't know, the folks at Neuren Pharmaceuticals, other than a medical monitor who is essentially sits in a data safety monitoring board that sit on the outside to be sure things are safe. No one knows who's getting what, so we can't essentially do an interim analysis to say, a certain functioning level or a certain age individual is doing better at this point. Really what will happen is at the end of the trial, when the data comes out, it kind of comes out in bulk, so to speak, and then we have all the information at one point.
Okay. So we're getting questions in. They want to know when it starts and when it ends. So do you want to give a little information on that?
As far as, well, the trial is active right now. So, the sites are up and enrolling. It's been ongoing for some time. I think with confidence as we sit here at the end of April that there are months left in enrollment. It's one of those things that if all of a sudden tons of folks enroll and slots are full, it's going to be done quicker. If it drags on because of, a slower pace of recruitment, it lasts longer. But, I think we're definitely, in my mind, looking at likely May, June, potentially longer for folks to get in. When I get asked that question in Cincinnati, because people call up and say, Dr. Erickson, how long are you enrolling? I always try to be honest and say, well, I can't control when the study's going to end because it's based upon when the slots are full. But I would tell people that called up our Cincinnati clinic right now that, likely throughout May, certainly, and likely throughout June, we're going to still be enrolling. Beyond that, it depends on, if slots are full.
And so if this is a 10-week study, and let's say that you're still taking people into June, do you have any idea when, the initial results would be out, or when we could expect to hear something from the study?
I mean, that's a great question, and I think, honestly, Jane, we should take that back to Neuren and provide their best estimate, because in some ways I can't speak on their behalf. But what I can say is, if you look at a rough timeline, and let's say the last individual is dosed on July 1st, you count 10 weeks from there, you're in, through July to August to September. It certainly takes time to analyze data, look at final safety data. We're definitely looking at on an optimistic side, potentially final quarter of this calendar year. But I would really defer to the folks at Neuren, and I think we should ask them, and they could probably provide a nice estimate for those on this webinar and in the Fragile X community.
Okay, I can work on that and get back to the group. So is this just for people with Fragile X syndrome, or will you include carriers of the premutation?
This is for individuals with Fragile X syndrome as defined by the classic, Fragile X PCR and Southern blot testing.
Okay, so greater than 200 repeats of CGG?
Absolutely, yes.
Okay, we have a question. Acamprosate is an excluding drug. How would one know if it would be beneficial to come off and try this medication?
It's a great question. It's a question that comes up, in our clinic, because, those that know me know that I've done development work with acamprosate. We have an acamprosate clinical trial in a little bit of a different age range in Cincinnati and at Rush Medical Center. And what I'll often tell families is, look, no matter what the drug is, if it's a new potential targeted treatment and your child's taking it and you feel they've improved on it, first of all, the only way, we can answer how long you need to continue one of these treatments, because we don't have great answers, is to potentially stop it and see what happens. You hate to be that sort of blunt, but it's the truth. So if a family asks me, my child's been on acamprosate for 2 years, they've made considerable gains, I'll counsel them in that way. I'll say, hey, if you want to consider stopping it, see what happens to the gains, be off of acamprosate or any other, medication, racemic baclofen, minocycline, lithium, you name it. If you want to try off of it and observe how things are going and then roll into a new investigational study, whether it's NNZ or anything else, then that's fine. The risks may be that any gains you had theoretically from another treatment could go away. I'll tell people specifically with acamprosate, when we've had people go off of it clinically for whatever reason and then they've gone back on it, if they lost gains, the gains tend to come back, at least anecdotally. But, we can't make that guarantee. But, I just have honest, frank conversations with families and say, we're not sure. I can't say exactly what will happen if you stop whatever treatment to get in the trial. And that's your choice. And the risks is some of the gains may go away, but you could certainly restart that treatment later. And I think there's high likelihood they may come back, but there are obviously no guarantees. So like anything in medicine, it becomes a risk-benefit analysis, and we're just going to provide the best information I have or another clinical trial site has before making that decision to make a change to enter a trial. And that really goes for acamprosate or any other of the exclusionary medicines that we mentioned tonight.
So do you think it would be best then if, if someone was considering this and had to go off another medication, just to talk to the doctor or whatever clinic they were going to that is participating in this study?
I think it makes great sense that if you're going to go off a medicine to enter a trial, to discuss it with whoever prescribed the medicine you may go off, and have a frank discussion with the clinical trial site. Because no one involved with this research wants folks doing something that would have a negative, significant clinical impact to enter a study. It's the kind of thing, and I'm not saying that's the case with NNZ at all, but if someone's having, significant aggressive behavior and they stop a medicine that I really think is helping that aggression to get in a trial, I'm going to have to say, man, maybe you should have second thoughts about that. But every case is different, so just have an open discussion. I don't ever want a family, and I think I can speak for the Fragile X research community, we don't ever want to be putting pressure on families to come off a treatment that they stand by and feels well tolerated and efficacious for their child to enter an investigational drug study. That's not the goal. If we're not sure what's going on or folks would like to try another option, by all means. But, folks should not be feeling pressure to try to, fill some entry criteria in that way. That's not the goal.
Well, so then along those lines, so at the end of the 10 weeks, is anyone able to stay on this medication or until it's feasible?
No, to be straight up, the answer is no. There's no current open label extension. There's some data in the animal model that positive gains in the mouse were maintained after stopping treatment in the mouse, but obviously there's no human data published yet available that shows continuation of effect. So there's no open label extension. When the trial's done, it's done. So we can make no guarantees about future access to the compound based upon current participation. It would all be conjecture at this point.
But is that a possibility? Do you know? I mean, I guess we can only conjecture. We'd have to go back to Neuren to see where that stands. Is it a possibility, though?
I think anything's possible. You like to look at past precedent. I mean, there have been past precedent where industry has developed open label studies, sometime after initial trials were completed and allowed folks in. So it's a possibility, but it's by no means a guarantee, and I can't speak for Neuren Pharmaceuticals in describing their intentions, and I also can't speak for the United States Food and Drug Administration and what they may allow a specific company to do in an individual case.
So someone did just ask, so why is it only males? We didn't ask. We asked if it was going to be any girls, but why is it? Why would you just start with the boys?
That's a great question. I think, there can be multiple factors. We can query Neuren for specifics, but generally, as a clinical researcher in the field, if you're involving just males, you get a little more refined population because the males kind of by definition are having deficient protein expression because they don't have the second X chromosome to potentially help cover. So it's a little more clear pathology. There may be aspects of safety and other data that led to that conclusion, but that's kind of another point that we should probably ask Neuren about and maybe post a more accurate, complete response because I'm sure they can answer better than I.
Make a note of that. So one of the questions, let me see if I can find this. It was, that you showed the test results in has the brain been measured in live humans the same way as it was measured in mice? My questions keep jumping around over here.
No, the answer to that is no. There's no kind of live human brain imaging that has looked at change. I mean, to be honest, the kind of brain change we talked about cellular connections changing, we don't really have the technology in living humans to study that, as you're living and breathing. We do have technology to look at aspects of blood cell activity that reflects some of these chemical abnormalities in Fragile X, and I believe, Neuren is going to look at that. But no, we can't really get an eye into, brain chemical and brain connections while folks are, alive taking a new medicine. We'd love if we could. Hopefully in my lifetime we can do stuff like that, but it's not on the table right now.
And so if people want to sign up, did we go back, we can post this list of all the places that are doing this trial. Would they just contact those sites directly?
Yeah, they should just contact the sites directly, and you can go to the clinicaltrials.gov website. Should give you contact information for the individual sites. And then this study website here, fragilexstudy.org, essentially, will also help provide those connections. So either clinicaltrials.gov and search NNZ and Fragile X, or the fragilexstudy.org should be able to get you connected with what may be the easiest location for potential participation.
Okay, and I just want you participants to know that if you're not sure where to go, you feel free to contact me on that too. Okay, so this is a trickier question. How are the results with the knockout mouse when you compare them with previous drugs that failed with humans?
That's obviously a great question. I mean, I'll kind of take it point by point. The brain connection differences. So when we showed, the fancy green pictures of how brain cell connections were normalizing with NNZ treatment, similar findings with selective type 5 glutamate receptor blockers, to a degree, some similar findings with an STX-209. Some of the chemical changes, in particular the ERK changes, similar to selective type 5 glutamate receptor blockers in the mouse. The AKT findings inside the cell, really less published data in some of the previous drugs, so less comparison there. Some of the behavioral changes in the assays that I talked about are a little different than what's been done and published. But, to be honest, I think the collective body of the mouse findings is somewhat similar to what we've seen with other compounds. And, I think the obvious unspoken part of that question is, hey, if these guys are putting out a portfolio that's similar of what's failed in humans, why do we pursue this one? And I think my answer as a clinician and a scientist is, well, we have so many different tests that we have been giving to mice in the field. I think we're evolving new tests. But, if you have a specific menu of tests and drug A, B, and C work, and then this is drug D and it works, don't penalize drug D for using the same menu of tests. It's sort of a limitation of our preclinical mouse science, and hopefully we develop better assays that are more aligned with human response. But I wouldn't, necessarily punish NNZ for that. What I would say is the mechanism of action of the drug is quite different and broader in a number of ways than some of these previous molecules. So that's where I think as a clinical scientist I see a real difference that warrants exploration, that it's not selectively hitting one glutamate or one GABA receptor. I think I take kind of more heed in that than I do sort of seeing this pattern of similar animal results in some ways.
Gotcha. Well, so along those same lines, a question came in. How is Neuren Pharma compared to Seaside? I mean, what are the chances, that will happen with what the Seaside could happen here? Or is it all test results? We would just see what kind of test results show.
I think we just don't know the answer. That's a predictive thing that we don't know. I think we're hopeful that the lessons, as I said initially, the lessons learned in clinical trial failures are not being repeated in the same way with Neuren and hopefully with other partners in the field, that we're taking a different approach to outcomes. We're taking some different approaches to study design to individualize the way we look at things to hopefully, more adequately detect a signal. And we're using a medicine that has a really different mechanism and potentially a broader mechanism of action. So none of us can guarantee the results of this NNZ trial. We don't know if it's going to move on to phase 3 larger studies and what's going to happen. I can just describe for everyone on this webinar, where the trial's at, how it's different from previous trials, how the mechanism is different, and how as scientists in the field, we think it's important to move forward other potential treatment options that have some hope and potential to change the outcome, though obviously we cannot make guarantees.
So do you know how many people, so so one of the questions, I know of someone who's already been through a 12-week trial of this med, so the trial that's coming up, so is this phase 2?
Neuren Pharmaceuticals has only had a single clinical trial in Fragile X, and that's the one ongoing. So if somebody's been in it, they were in it a while ago. I mean, it's been ongoing for a time. I don't want people to think it just started and it's going to be done in a couple of months. It's been going on for many months, so you may well know someone that was in it X number of months ago, but it's all the same thing. There's not sort of like a first NNZ trial in Fragile X, and now we're on the second one. There's only been one, and that's the one we talked about this evening.
So do you know what their target number of people, number of people to be in the trial is, and where they stand right now?
I believe it was about 60. They may be expanding the number, so that's something that I don't want to misstate, and I think clearly we should follow this up with a Neuren Q&A posting. We can get those exact numbers. Are they expanding? When's it anticipated to be done? Where are the numbers at? I think that can be provided. But we're certainly, if this was a baseball game, we're not in the 9th inning, but we're in the 6th, 7th, 8th, second half of the ball game on the project.
Some questions are starting to come in about other medications, and I don't know whether you want to take those or if we want to just wait and have another webinar with you where you do your medication webinar.
I think sometimes, Jane, I should just tune in from home and just take Q&A for 60 minutes would be the way to do it. I may get in trouble doing that, but I'm happy to answer. Let me look at the screen here.
The very last one. What medication is better or not?
That's a $25,000 question. I can tell you what's in development. I mean, Alcobra Pharmaceuticals has recently completed recruitment in a molecule they're studying more for ADHD symptomatology associated with Fragile X. We've talked about NNZ. We do work with the Camprosate. Rondi Hagerman's doing work with ganaxolone, a GABA modulator. There continues to be interest in minocycline. I have to say, I don't think at least since I've last done one of these webinars, there haven't been a lot of, recent breakthrough publications where I can say, oh my gosh, something's ramping up for a new trial that's going to hit in the next couple of months that I haven't already mentioned. So I think that's kind of a general response, but I don't have a better answer.
Maybe as a final note, I forwarded you, there was it was on the Google alert came out today about the Seaside trial, or the medication that was used there that was shown to fail in Fragile X syndrome, but they're maybe looking at using it, repurposing it for people with autism. Do you have any comments on that? The results of that article that I sent you right before the start of this webinar?
Yeah, I think, scientifically what came out was, some preclinical work with R-baclofen that showed that potentially some of the gains in social behavior that were noted over time, our baclofen treatment may be causing in animal models upregulation or additional overactivity of type 5 glutamate receptors. So basically that may be a reason why folks could have reported some short-term gains and over time the effect wore off. So I think that's incredibly interesting to explore and may help provide some explanations. What was also written on the Simons Foundation, and the Simons Foundation is a very highly regarded autism philanthropic funding and science organization, was that there may be some interest in the Simons Foundation funding STX-209, our baclofen study in autism. I don't think we know if that would include individuals with Fragile X syndrome. I think there's a fair chance it may not. But I'm personally going to make an effort to contact the Simons Foundation. If I can get any additional information, we'll certainly pass it on to Fragile X stakeholders via the foundation if we get more information on that, because I think it's important, because I know so many families have been involved with Seaside and with STX-209 development and trials. And if there are other opportunities or other directions, we want to get that word out and provide that information.
Okay. That would be great. And, yeah, and I can work with you on that. Let's take one last question, and which is kind of an interesting one. You know, is there a medication out there on the market now that mimics any of what the NNZ is designed to do? You know, like an over-the-counter medication or something that can be prescribed. You know, kind of similar, I think, along the minocycline that was originally designed for acne. You know, is there anything else out there that mimics what NNZ does? That we
No, there's no real clear mimics, because, you know, it's a synthetic analog of this, you know, product of insulin growth factor activity. There's nothing really like it that's available, and I wouldn't advise individuals to go off and try to find, you know, insulin growth factor itself in different ways. I think that, you know, this is a much more targeted and clean, developed, safe approach. So there's nothing that I know of that's a rough equivalent to trial.
Okay. Well, I want everyone to know we're going to cut it off here. It's after the time, and but we will have Dr. Erickson back for general medication questions, probably in the next early mid-fall this year. He's agreed. If he hasn't already agreed, I think we'll volunteer.
I'm a regular on these webinars. You know, this one worked out really well. The IT was perfect, and, you know, I just kind of sit at home. Maybe you hear my kids yelling once or twice. I have a good time. I love doing it, so happy to continue.
That's great. We sure appreciate it. Yeah, next time you should bring the kids on so we can see them and, you know, make sure everyone.
I certainly will. They'll enjoy the opportunity. So.
Okay, let's see. You know what? So, Craig, can we just throw in one last question? I see this now.
Sure.
Any plans for young boys with Fragile X? Our child is 3.5 years old. Any trials? And if not, why not?
That's a great question. I mean, NNZ, I said, made it go down to age 12. You know, I'm working with researchers, you know, around the country. Liz Berry-Kravis is really leading the charge with others to potentially bring previously studied medicines that failed in older age groups into younger children, because part of the big question is, how early do we need to treat to really move the needle and make a difference? We wouldn't want to miss a medicine that could be incredibly transformative for toddlers because we didn't see a huge effect in adults. So I think that we're taking those efforts and trying to do that. We should have word by the latter part of this year if some of our researchers in the field are able to do that with medicines that have already been studied in older adults. The real reasoning is kind of a safety reason, that these medicines are often developed first with adult safety data, and then sort of trickle into younger age ranges. And that's the United States Food and Drug Administration's way of saying, hey, best to try in adults first, because if we study safety in adults, including adults without the disorder, like adults with otherwise normal development, they're able to describe how they're feeling. They're able to describe if they're having any ill effects. Once you prove something safe in adults, then you move into younger age ranges. But you can just imagine doing a safety study in toddlers is exponentially more challenging, has more ethical issues surrounding doing safety studies in adults. So that's the reason why there's a delay. But I think the individual that asked that question and everybody on this webinar can understand that we as a Fragile X research community get that message, that we want to go younger when possible, when safety data is there. We want to generate the safety data if we need to do it, because it really resonates with us that, hey, Fragile X is a lifelong disorder, and the earlier the better. So hopefully we have some positive news in the next 6 to 9 months about those efforts, but I'll tell you we're trying.
Great. Well, thank you. Thank you, Dr. Erickson, for sharing your expertise with us. You're a wealth of information. So we'll just plan. I want everyone to plan maybe September, October. We'll work with Dr. Erickson to set up a time for just where you can ask all your questions about medication, and you can get some answers, and maybe we'll have, who knows, maybe we'll have some results from the Neuren study by then. But we will work with Neuren to get answers to some of your questions that brought up that we didn't have answers to, and we'll get that out. We'll send an email out on that. So thank you, Dr. Erickson. We sure appreciate your time.
You're welcome. Good night, everybody.
And I want to tell people in the next webinar, we're going to go back to the Let's Talk series, which is for members only, and we're going to have Dr. Rondi Hagerman discussing premutation carrier issues. It will be scheduled probably the last week, sometime in the last week of June, so watch for email announcements about that. So we're signing off. Again, we'd like to thank Warren Global Conferencing for the webinar tonight. Have a wonderful evening, and if you have any questions, don't hesitate to email me. Good night, everyone.